Good afternoon, learners. Welcome you all to this Gyan Darshan session. We all know by this time that the MSc Zoology program has been launched in January 2024, and semester 1 and semester 2 already we had one term end examination in December. Now in semester 1 there is one course. By this time you are familiar. We have a course on molecular cell biology, MSCZOO 001, and it has 4 blocks consisting of 16 units, and in unit 16 there is one topic, that is cell death and renewal. So today we will be focusing on this unit, that is unit 16, cell death and renewal. Cell death, which is also otherwise termed as apoptosis, which is more specifically the programmed cell death, okay, and the renewal of a cell. So these are the two topics that we will be discussing. Now, what is meant by programmed cell death or apoptosis? See, whenever a cell suffers any injury, then for the protection, for the overall protection of the body, the cell has to be destroyed. Now, if there is an accidental injury, we don't call it apoptosis because the cell death is not programmed. It is just the destruction of the cell for the protection, and we call it necrosis. It is not apoptosis. Apoptosis is a more regulated and organized kind of phenomenon, and it involves a lot of molecular biological events, the cell signaling events, a lot of proteins, and a lot of apoptotic regulators, pro-apoptotic regulators, which today we will discuss. Now in the first slide you can see that apoptosis or the programmed cell death, how will you identify that a cell is undergoing apoptosis? So first of all, we have to understand what is the difference of a cell that suffers death due to accidental injury, and what are the symptoms of a cell showing apoptosis. So these two differences you should be able to morphologically differentiate between these two types of cells. That is, one cell suffering an accidental death and another cell which is suffering from apoptosis, or not suffering, which is facing apoptosis or programmed cell death. So if you see this particular slide, that is apoptosis or the programmed cell death, you see a normal cell. Then this normal cell, it shows certain symptoms like cell shrinkage, chromatin condensation, membrane blebbing, nuclear collapse, apoptotic body formation, and then the lysis of the apoptotic bodies. So these are the different signs or the different features of a cell that undergoes apoptosis. So here you can see that again in this particular slide you can see that a viable cell, there are two options. One, it can pass through apoptotic stages, and another, it can pass through the necrotic stages. You can see that there is a basic difference. In case of apoptosis, there is a shrinkage and chromatin condensation, as we have already seen in the last slide. Then budding, membrane blebbing or budding, apoptotic bodies phagocytosed with no inflammation. So one major difference between a apoptotic cell and a necrotic cell is that in case of necrotic cell, it undergoes inflammation. The cell swells or it explodes, and it releases certain toxic materials to the exterior of the cell. Whereas in the apoptotic cell, it's just the reverse, that is, the cell undergoes shrinkage, the cell undergoes condensation, whereas in a necrotic cell, it undergoes swelling. So this is the major difference. So microscope, when these two kinds of cells are placed in a microscope, You can, in an electron microscope, if you are asked to examine or distinguish between these two cells, so this is the first sign that will help you to distinguish between these two types of cells. So, necrosis is a passive cell death, exhaustion of oxygen or nutrient, exhaustion of ATP in the cell, impairment of the cell membrane, and enzyme release causes inflammation. Whereas if you see apoptosis, here also it is cell death, but there are a series of intracellular signaling which we will discuss today. Fragmentation of the cell into vesicles and phagocytosis by the neighboring cells. So apoptosis is a more organized and well programmed event, whereas necrosis is a very random kind of event. So this is another difference. That is why we call it programmed cell death, because the death of the cell is programmed. It is very much organized or regulated. Whereas in case of necrosis or the accidental cell injury, it is very random. It is not well regulated or well organized. So morphologically, what are the following stages that will enable you to distinguish? As we have discussed, volume of the targeted cell decreases. It does not expands. There is cytosolic shrinkage, chromatin marginization, nuclear condensation, redistribution of the nucleus fragments to blebs on the apoptotic cell surface, cytoplasmic blebbing, Solene endoplasmic reticulum. On occasion, mitochondria normal or condensed. One enzyme called transglutaminase, it creates nets, and so apoptotic bodies can be phagocytosed. So there are several terminologies we have come across, particularly apoptotic bodies. We will come, when we discuss the entire series of events, we will come across these terms one by one. So see this electron micrograph. So in the first, that is numbered A, this is an example of a necrotic cell. So see, there is an explosion, or the cell has expanded and it has released all the toxic materials outside. So there is a swelling of the cell. Whereas in case of number B or number C, if you see the figures, then the cells are more condensed. They are not exploded or they are not expanded or swollen. Rather, it's just the opposite. That is, the cells are condensed. So B and C, they represent the apoptotic cell, whereas A, it represents a necrotic cell. So this is the basic difference. Now we come to the next point, that once you have seen how to distinguish between an apoptotic cell and normal cell, the next question is then why the cell undergoes apoptosis? Why the cell intentionally undergoes death? Because cell death, we know that a cell dies because of some injury. Whenever we face any injury in any part of our body, human or any animal, then we try to remove that destroyed cell, because otherwise the healthy cells will get infected. So we try to remove that injured cells from the body, and that is why the cell dies. Or the necrotic cell, suppose you face an accidental injury. So there is a cell injury. So then because of that injury the cell will die. But if you consider that a cancer cell, so if the cancer cell spreads across the body, then what will happen? Then the entire individual will become cancerous and there is a scope of death. So if there is any mechanism in our body which will help to kill the cancer cell, then our body can be protected. That will be a defense so that the cancer cell does not spread to the surrounding tissue. So the cell should have some mechanism by which it can kill the cancer cell. If it does successfully, then our body is saved and we will survive. So this is an apoptosis. That is, it helps you, this kind of cell death actually helps you in your survival. So we will discuss this. Now here in this slide you can see two examples. When a tadpole, it changes into a frog. What is the basic difference in the morphology between a tadpole and a frog? A tadpole, there is a tail, you can see the tadpole tail is there. But in frog, it does not have a tail. So this is called metamorphosis. When the tadpole, it converts into the adult frog. But in this process, the tail disappears. How it disappears? It disappears through programmed cell death or apoptosis. Sometimes you might have noticed some human individuals, we have 5 fingers or 5 digits or 5 toes. So sometimes you might see that in some individual there is an additional finger, additional digit formed, maybe in between the thumb and the first finger. There is an additional digit formed or small kind of finger generated. How? Because these persons are compromised in apoptosis. Because our body is trying to divide, the cell of a normal individual is trying to divide every time. But apoptosis keeps them suspended. And that is why in normal individual like us, we do not have that extra digit or extra finger. We have only 5 fingers. But if this apoptosis mechanism is disturbed or suspended due to some reasons, then it might happen that there can be an additional finger formation. Because in these individuals, the apoptosis is weak. That is why the cell death is not occurring. And that is why the cells are dividing and they are giving rise to some extra tissues, unwanted tissues. So I hope you can understand what is the significance of apoptosis. It helps to maintain the normal condition of an individual. Whether in terms of non-cancerous cell, whether in terms of having the exact number of tissues in our body or the presence of normal organs in our body. So these are very important. So significance, balancing cell proliferation and maintaining constant cell number, because our body, in our body, the cell is constantly dividing, constantly cell division is occurring. And so the number of cells that are produced are always in excess than the number of cells which are actually required. Then those excess cells, how will you remove those excess cells? It is through cell death or it is through apoptosis. The nerve cells, the red blood cells, all these cells are produced in much excess than what is actually required. So these excess cells will be eliminated from the body through apoptosis or programmed cell death. So if apoptosis was not there, there would have been a tremendously higher number of cells. And so there might have arisen several abnormalities, like in some Parkinson's disease. The number of neurons are much in excess than that which are actually required. So there is a miscoordination. So also this is true for the red blood cells. So it provides a defense mechanism by which damaged and potentially dangerous cells can be eliminated. Virus infected cells, they undergo programmed cell death by preventing the production of new virus particles and limiting the spread of the virus. So they try to keep the virus restricted within some specific cells so that it does not spread to the surrounding cells. In case of DNA damage, because DNA, it undergoes damage due to several reasons, due to different genotoxic stress. So there the programmed cell death can eliminate cells carrying harmful mutation. Unwanted cell, I already discussed, like larval tissues during insect metamorphosis, tissues between digits during finger and toe formation. These are eliminated during apoptosis. So overall, apoptosis helps in maintaining proper association, proper coordination, proper communication between the cells and tissues. So it is a very, very significant event in maintaining the normal condition of an individual. So I hope you understand why the apoptosis is required. What is the significance of apoptosis? First, we learned how to identify an apoptotic cell. Second point, we learned what is the significance of apoptotic mechanism. Now we will go into more details about how this phenomenon occurs. Okay, what are the events that trigger programmed cell death? Neurons, as I said, neurons are produced in excess. Up to 50% of the developing neurons are eliminated by programmed cell death. Those that survive are selected and make proper connection. But if all of them would have survived, what happened? What would have happened? There would have been miscommunication between the neurons. So there would have been a miscoordination. Okay. So how does PCD differ from accidental cell growth or accidental cell death? So in accidental cell death, there is acute injury, cells swell and lyse, release their contents into extracellular space causing inflammation. PCD or programmed cell death, abbreviation for programmed cell death or apoptosis, is an active process which proceeds by a series of cellular change called apoptosis. Chromosomal DNA is fragmented due to cleavage between nucleosomes. So chromosome fragmentation, chromosomal DNA fragmentation, this is another sign of understanding or identifying an apoptotic cell. Another very important symptom, I won't say symptom, sign, another very important sign of identifying an apoptotic cell is normally the phosphatidylserine. This is one of the major plasma membrane component. Normally this phosphatidylserine is localized in the inner leaflet of the plasma membrane. But for an apoptotic cell, it is exposed on the cell surface. So the distribution of phosphatidylserine will also help you to distinguish between a normal cell and an apoptotic cell. Now we will take mostly two organisms. One is Caenorhabditis elegans. What are the components which regulate apoptosis? And then we will come to mammals, particularly in humans, that how this phenomenon, we will read the mammalian apoptosis in greater details, but we are also studying apoptosis in Caenorhabditis elegans because it was first identified. The genes for apoptosis were first identified in Caenorhabditis elegans, and later on the study was further extrapolated in humans or mammals. So in Caenorhabditis elegans, initially CED3 and CD4, these are the two genes, cell death abnormal, the full form. CD3 and CD4, these were the genes. So the scientists found that if these two genes are mutated or if they are inactivated, the programmed cell death will not occur, apoptosis will not occur. And there is another gene called CED9, which is a negative regulator of apoptosis. So if CED9 is active, then what will happen? Apoptosis will not occur. If CED9 is inactivated, then apoptosis will occur. So it is a negative regulator. So CED9 on inactivation, the cells that would normally survive fail to do so. Instead, they undergo apoptosis. But if CED9 is expressed at abnormally high level, then there is no programmed cell death. So, of course, you have to understand that in a normal cell, there is no apoptosis because in normal cell you don't need apoptosis. Why should it undergo cell death? There is no need. So, in this case, if there is no PCD, you have to understand that CED9 is present at a very high level, and therefore CED3 and CED4 are inactive, because if CED9 is active, then CED3 and CED4 should be inactive and vice versa. Okay, so you understand the relation between or among CED3, CED4 and CED9, of which CED9 is the negative regulator, CED3 and CED4 are positive regulators. So CD4 is actually helping in stimulating CD3. So CD3 is probably the main player. And CD3 is activated by CD4, whereas CD9, it inhibits CD4. So CD9 is a negative regulator, CD4 is a positive regulator, but it further stimulates CD3 and then CD3 works. So CD4 is present in the upstream part, upstream signaling, and CD3 is present in the downstream signaling. Now what is this CD3? I said that CD3 is a gene that triggers apoptosis. So if it is a gene, then it should encode a protein, because we know that a gene encodes a protein. So if CD3, it encodes one protease. That means an enzyme which degrades a protein. So it encodes a protease. And these proteases are called caspases. So apoptosis is actually regulated or controlled by a series of proteins or proteases, enzyme proteins, proteases called caspases. Caspases, they have cysteine at their active sites and they target any protein after aspartic acid residues. So they destroy any protein by cleaving after aspartic acid residues. But these proteases, they have cysteine residues at their active sites. So cysteine is a vital amino acid in caspases. So if they destroy, these caspases, they can also act on the target proteins, as I said, and they cleave the nuclear lamins, leading to the fragmentation of the nucleus, cytoskeletal proteins, and also they disrupt the DNA, the nucleic acids. They lead to the disruption of the cytoskeleton, membrane blebbing, and cell fragmentation. So all the signs that we have seen in an apoptotic cell, they are triggered by caspases. Now in Caenorhabditis elegans, CD3 is the only known caspase. In Drosophila and mammals, at least 7 caspases are there. Now we will discuss how these caspases are activated. I mean, whenever a cell has to undergo apoptosis, then these caspases, they have to be activated, because the caspase remains in an inactive state in the body. In the normal cell, the caspases are there, but the caspases remain in an inactive form, which are called procaspases. And these procaspases, they need to be activated through some proteolytic cleavage, catalyzed by other caspases. So there are two groups of caspases. One is initiator caspases and another is the actual caspases or the activator caspases. So initiator and then the effector. So one who initiates the process, how do they initiate the process? These initiator caspases, they actually undergo proteolytic cleavage of the target caspases or the effector caspases, and then the effector caspases get activated and then they start working. Then they start the further actions to trigger the programmed cell death. Now in mammalian cell, caspase 9 is the key initiator caspase. It gets activated by binding to APAF1, which is a mammalian homologue of CD4 in a multi-subunit complex called apoptosome. So apoptosome formation is one very important feature or event during apoptosis. This complex formation requires cytochrome C, which is released from the mitochondria by the apoptotic signal. So whenever a signal is received, one very important feature is the cytochrome C is released from the mitochondria. Normally cytochrome C is present within the mitochondria, but during apoptosis this cytochrome C is released and there is the apoptosome complex formation. After being activated, caspase 9 initiates proteolytic cleavage of procaspase 3 to form active caspase 3. So procaspase 3 is inactive precursor, caspase 3 is the effector caspase. And who is the initiator caspase? Initiator caspase is caspase 9, because caspase 9 actually helps in the proteolytic cleavage of procaspase 3 to caspase 3. Only then the cleavage of the nuclear lamins and all other events that we discussed, it will occur. Now in the next slide you can see the exact the diagram. If you see the diagram, you will understand now. In the left hand side, if you see the diagram, procaspase activation. So initially there is an inactive procaspase. There is a cleavage site and therefore the pro domain is being released. So it's activation by cleavage, and then you have the active caspase. And then what you see is very interesting, that it's called a caspase cascade, because whenever you generate one active caspase, it will bring many molecules of active caspase together. And it's a kind of chain reaction or amplification reaction or chain reaction. And several active caspase molecules, they get assembled. And more caspase molecules, they are aggregated or assembled, giving rise to the series of events like chromatin marginization, whatever we discussed, nuclear condensation, shrinkage, cytoplasmic shrinkage, membrane blebbing. So all these symptoms. So what are the learners? What did we learn? We first learnt about the caspase. We learnt about the two types of caspases, that is initiator caspase and effector caspase. Initiator caspase, what is its main function? Its main function is to trigger the active, is to activate the effector caspase. How it does so? It first undergoes proteolytic cleavage of an inactive procaspase to active caspase, and then what happens is a chain reaction. Several active caspase molecules, they are assembled and aggregated, giving rise to the further downstream signaling events. So this is the event. So if we go through step by step now, that is, activation is triggered by adapter proteins or initiator procaspase. Initiator procaspase have a small amount of protease activity, forcing them into a complex tissue formation, assemble them through mutual activation. So each of them, they mutually trigger proteolytic cleavage and they mutually activate each other. And this aggregation, it causes a conformational change that activates the procaspase. So this assemblage or this aggregation is very, very important to trigger the proteolytic cleavage and to trigger or activate the procaspase into active caspase. So if the function of the caspase and the activation of caspase is clear, now we will bring into this picture other events, or we will bring into this picture other candidates or other regulators. So accordingly, we can divide the apoptotic regulators into two groups: the anti-apoptotic regulators and the pro-apoptotic regulators. Anti-apoptotic regulator means those regulators which will inhibit apoptosis, and pro-apoptotic regulator means those which will activate apoptosis, or those which will favor or positively regulate the apoptotic mechanism. Now, what are the anti-apoptotic proteins? Anti-apoptotic proteins, they are mostly known as the BCL2 family proteins. Initially, this BCL2 was identified as an oncogene. Oncogene means a gene which triggers cancer formation. So you must be understanding that a cancerous cell and an apoptotic cell, these are just the reverse. If a cell is cancerous, there will be no apoptosis. If a cell undergoes apoptosis, obviously there will be lesser chance or no chance of cancer formation. Why we are healthy? Why we are non-cancerous? Because the apoptosis is going fine. But if the apoptosis mechanism is affected, the chances are that the cells may turn into cancerous state. So you see how this apoptosis is saving us, defending us from cancer. So BCL2 is an anti-apoptotic gene, and therefore in a normal cell, okay, BCL2 is not present or is not activated. In an apoptotic cell also, the BCL2 will be not activated. Okay, because the pro-apoptotic proteins, which are the pro-apoptotic proteins? They are again divided into three groups. The pro-apoptotic, not three groups, two groups, pro-apoptotic multi-domain and pro-apoptotic single domain. Pro-apoptotic multi-domain, BAX and BAC, they have three homology domains, BH1 to BH3, and pro-apoptotic single domain, BID, BAT, NOXA, PUMA, BEAM, these are the different proteins. So all of them are the pro-apoptotic proteins. So if you will see now, if we try to assimilate or if we try to assemble the entire picture, we will be coming shortly that the entire picture how it occurs. But before that, we are just focusing attention on which are the regulators. So mitochondrial pathway of apoptosis, as a result of damage to mitochondria, apoptosis is induced. When activated, the pro-apoptotic BCL2 family proteins form oligomers in the mitochondrial outer membrane. Therefore, the cytochrome C is released from the mitochondrial membrane, and then the release of cytochrome C to the cytosol followed by its binding to APF1, which triggers apoptosome formation and caspase 9 activation. So when caspase 9 is activated, after that the caspase 3 will be activated, because caspase 9 is the initiator caspase. But even to activate the initiator caspase, you need some other upstream events which we have discussed now. That the main signal should come from the mitochondria through the apoptosome formation and the release of cytochrome C from the mitochondria. So caspases are also regulated by a family of proteins called IAP, inhibitor of apoptosis family. IAP members directly interact with caspase and suppress apoptosis. So inhibitor of apoptosis, that means which will inhibit apoptosis. How do they inhibit apoptosis? They suppress apoptosis by inhibiting caspase activity or by targeting caspases for ubiquitination and degradation. Ubiquitination is a form of destruction of the proteins. So if the caspases get destroyed or degraded, then there will be no apoptosis. IAP does exactly that. And that's why they are called inhibitors of apoptosis formation. In mammalian cells, there are yet other proteins that promote apoptosis, like SMAC, Diablo, OMI, HTR2, that stimulate cell death by interfering with the action of IAP. So again, there are proteins which will try to inhibit IAP. So if you can inhibit IAP, then you can promote apoptosis. SMAC, Diablo, these are some pro-apoptotic proteins which try to suspend IAP and thereby promote apoptosis. So it's just a tug of war going on between a anti-apoptotic protein and a pro-apoptotic protein. Which will win? So there is a tug of war. There is a race going on between these two groups of proteins. So BCL2 family proteins and IAP proteins are the main intracellular regulators of cell death program. So I hope it is clear what are IAPs. The IAPs, they inhibit apoptosis in two ways. They bind to some procaspase to prevent their activation, and they bind to caspases to inhibit their activity. So this is the main way or mechanism by which the IAPs, they stop the caspase activation. When mitochondria release cytochrome C, they also release a protein that block IAPs. So if you have to undergo apoptosis, then you have to arrest IAP activation. So you need again some proteins that will stop IAP. So that actually happens during the release of cytochrome C. So that is why release of cytochrome C is one very major event in the apoptotic mechanism. Now, whenever a cell undergoes apoptosis, then or let's say a cell, it has faced DNA damage. So if the cell still replicates, if the DNA still replicates, then what will happen? The damage will amplify. Supposing in your home a cloth is torn. Okay, a piece of cloth is torn, and you are using that cloth without stitching it. What will happen? That will increase, and at some point of time, that cloth will become unusable totally. So what you have to do? You have to stop using it. You have to sew it. Give it to a tailor, maybe, to sew it, and then use it. Similarly, it's the same for DNA. If there is a damage in the DNA, and if the DNA still replicates, multiplies, then this damage will go on in the successive cell generations, and the damage will amplify. So, the cell cycle has to be stopped, and that damage has to be replaced, or that damage has to be removed or repaired. In biology, we call it DNA repair. So, there are different repair mechanisms. So, there are two options. Either you can repair it and then the DNA replicates, or what you can do is you can just destroy the cell. What you can do is, if there is a torn in the cloth, you can totally discard the cloth. You have that option. You will not use it at all. That option also you have without giving it to the tailor. Here also, you just destroy the cell. There is a DNA damage. You don't allow it to replicate. You just kill the cell. How will you kill the cell? Apoptosis. So, you can understand that here also again, so it will involve a series of same proteins that we discussed earlier. Here you need some proteins called some proteins which are involved in the cell cycle, like P53. P53 is also called a tumor suppressor gene. So, P53 is again another component that will help in removing or downregulating the oncogenes. Tumor suppressor, which suppresses tumor formation, which suppresses cancer formation. So, this P53 will also promote apoptosis. Someone who is stopping the oncogene or the cancer promoting gene will also stimulate apoptosis, because cancers and apoptosis are just the reverse kind of events. So, in these cases where there is a DNA damage and you need to totally kill the cell, then there is a way by which you can do, that is activation of P53, transcriptional activation of genes encoding BH3 only pro-apoptotic BCL2 family members such as Puma and Noxa, and that is why Puma and Noxa we call it as the pro-apoptotic proteins. In addition, there is another pathway called the tyrosine kinase pathway, where you have the activation of tyrosine kinase activated by PI3 kinase, which activates PI3 kinase. PI3 kinase phosphorylates the membrane phospholipid PIP2 to PIP3. PIP3, it activates serine threonine kinase called AKT, and then AKT then phosphorylates other proteins that regulate apoptosis. So, apoptosis is also controlled through phosphorylation of several proteins, and that phosphorylation, it occurs through this AKT. The key substrate for AKT is the pro-apoptotic BH3 only protein BAD. So, BAD is a pro-apoptotic protein that is phosphorylated by AKT. And this phosphorylation, it creates a binding site for 1433 chaperone protein that sequester BAD and maintain it in an inactive form. BAD phosphorylation, it inhibits apoptosis and promotes cell survival. So, the status of phosphorylation dictates whether a cell will survive or whether a cell will undergo apoptosis. There are other targets of AKT, FOXO transcription factor phosphorylation. It creates a binding site for 1433 proteins which sequester FOXO in an inactive form. In absence of AKT, FOXO is released from 1433 and translocates to the nucleus, stimulating transcription of the pro-apoptotic genes, that is BIM. So in that way, there are several target proteins of AKT. AKT can target several proteins, and the main task of AKT is phosphorylation of the target proteins. Now, so long what we have discussed is essentially the intrinsic pathway of apoptosis. What is intrinsic pathway? Intrinsic pathway means what is happening within the cell, that is within the plasma membrane. But the actual signal comes from outside. So, the plasma membrane also has a role to play to trigger the cytoplasmic events. So, the signal that comes from outside constitute the extrinsic pathway of apoptosis. So, the intrinsic pathway and the extrinsic pathway of apoptosis are integrated, and that is why my next slide shows the two combined together, that is the intrinsic pathway and the extrinsic pathway. For the intrinsic pathway, you can see now within the cytoplasm the events. What is the main event which we have already learned? You see the mitochondria, okay, and from the mitochondria you can see the release of cytochrome C. That cytochrome C, it gets combined with APF1 and caspase 9. APF1 to form the apoptosome, and that apoptosome activates caspase 9, and that caspase 9 activates caspase 3, and that caspase 3 promotes apoptosis through DNA fragmentation. And in between you have SMAC diablo and some pro-apoptotic factors, IAP, etc., which inhibit apoptosis, BCL2 which inhibits apoptosis. The arrows with pointed head means activation, and the arrows with blunt head indicates inactivation. Like BCL2, it inactivates. Now, the extrinsic pathway. Now we will come to the extrinsic pathway. If you go back to one of my slides, particularly if you take a look at this slide, here also we are just focusing on the intrinsic pathway of apoptosis. What is happening within the cell? Mitochondria, cytochrome C released, APF1, caspase 9, caspase 3, and then apoptosis. In between IAP, it inhibits apoptosis. P53 it promotes, BCL2 it inhibits. Now, what about the extrinsic pathway? Now, when you talk about the extrinsic pathway, you have to take a look at the membrane. In the membrane, you have certain receptors, which are called the FAS receptors. The one which is marked in magenta color, okay, that is actually the FAS receptor, or we call it FLR. And TNFR is the tumor necrosis factor receptor. Okay, but these receptors are present in the plasma membrane. And in the plasma membrane also, we find some changes. One change we have discussed about the distribution of phosphatidylserine. But here what happens is, you can see that there is some complex formation. You can see FLR, FADD, procaspase 8. So there is some complex formation. Similarly, TNFR, TRADD, procaspase 8. So there is a complex formation, and this complex formation is known as the disc, DISC. And this disc formation actually will activate caspase 8. That caspase 8 will activate directly caspase 3, or it will lead to the activation of a pro-apoptotic protein called BID. And that BID undergoes proteolytic cleavage to give rise to truncated BID or T-BID. And that truncated T-BID formation, it creates a signal for the release of cytochrome C from the mitochondria, leading to the apoptosome formation. So there are two ways by which caspase 3 can be activated. Caspase 3 can be activated directly from caspase 8, or the caspase 3 can be activated through another pathway where caspase 8 activates BID to T-BID, and then there is cytochrome C release, and that cytochrome C release lead to the apoptosome formation, leading to the activation of caspase 9 followed by caspase 3. Any of these two might happen, or in fact the two can occur side by side, hand in hand. So in Fas signaling, the Fas L stands for ligand, binds to Fas, leading to the receptor trimerization. Adapter proteins via their DDs, Fas associated dead domain protein, then bind to the cytosolic dead domains of Fas. FADD in addition contains a death effector domain DED to which the DED or procaspase 8 can bind. So you see that there is a complex formation between Fas receptor, Fas ligand. Ligand means the signal that is coming from outside. FADD and procaspase 8. This complex is called disc. After the disc formation, then we found two pathways. One is caspase 8 activation, directly activating caspase 3, or via BID. That is, caspase 8 can cleave the BID protein, leading to the truncated BID, which moves to the mitochondria, induces the release of cytochrome C release, and then the apoptosome formation, followed by the activation of caspase 9 and caspase 3. And the same event also holds true for the TNF receptors, just like the Fas receptors. That is, trimerization of the receptors followed by the same series of events. So I am not repeating the event occurring for the TNFR, but the nature of events are the same. Now another secondary messenger which is essential for Fas-induced apoptosis is ceramide. How is this ceramide generated in an apoptotic cell? Ceramide can be produced by the action of an enzyme called sphingomyelinase. Sphingomyelin is a very important component in the plasma membrane. In an apoptotic cell, there is action or there is activation of the sphingomyelinase, which hydrolyzes or degrades the sphingomyelin which is present in the plasma membrane to form ceramide. So apoptotic cell usually have a very high level of ceramide. So ceramide can induce apoptosis. Therefore, it has been found that many apoptosis inducing drugs, they increase ceramide concentration. Why? Because they want to promote apoptosis by increasing the level of ceramide, like etoposide, dexamethasone. These are some of the drugs which stimulate sphingomyelinase action. Daunorubicin, this is another drug which appears to stimulate the sphingomyelinase again. So if you want to kill any cell, or if you want to undertake programmed cell death, then you can induce this drug, you can apply these drugs, because they will induce the sphingomyelinase action, and therefore they will degrade the sphingomyelin in the plasma membrane, and the apoptotic cell death will occur. So that is all about the events, biological events or the cell signaling events, which I have tried to give you in a glimpse of the events in a short time. So you will mostly focus on the extrinsic and intrinsic pathways of apoptosis, the different caspases, how the caspases are activated, what are anti-apoptotic proteins and pro-apoptotic proteins, discuss the intrinsic pathway of apoptosis in details, discuss the extrinsic pathway of apoptosis, and try to coordinate between the extrinsic pathway and intrinsic pathway of apoptosis. So these are the things which you should learn. And what are the role of AKT and IAPs in regulating the anti-apoptotic proteins and the pro-apoptotic proteins, and also by and large the caspases. Now before we end today's discussion, I will take two more topics. One is, how will you measure the cell death? How will you measure the apoptotic cell death? Is there any way by which you can measure the extent of apoptosis? There are 2 or 3 assays. One is lactate dehydrogenase assay. So lactate dehydrogenase is present in the cytoplasm. When the cells undergo apoptosis, necrosis or any other damage, the plasma membrane is disrupted and lactate dehydrogenase is released into the cell culture medium. What is the function of LDH? It converts lactate to pyruvate, converts NAD plus to NADH. The NADH which is produced reduces a yellow tetrazolium salt into a water soluble red formazan dye, and this you can detect. How you can detect? You can measure it by calculating the absorbance at 492 nanometer using a spectrophotometer. What is the amount of formazan that is produced? So that will be proportional to the LDH present in the culture medium, and therefore this will be correlating to the number of dead cells or damaged cells. So that is the principle. Second is, you can do this analysis through fluorescence and non-fluorescence microscopy also. You can use certain fluorescent stains like DAPI, propydium iodide, ethidium bromide, acridine orange. You can use some non-fluorescent stain, methyl green, trypan blue. So these are some of the fluorescent and non-fluorescent stains which you can use to detect the DNA fragmentation or the DNA strand breaks. Because we have seen that DNA fragmentation is a very important sign or signal for an apoptotic cell. So you can measure this DNA damage or DNA fragmentation using these stains. Then there is one technique called Comet assay. This Comet assay is actually also given in your laboratory course 1. There is one experiment also which I have included in your laboratory course 1, MSCZOO 001. So Comet assay, what is the principle? Probably you have gone through the technique, but what is the principle? The cells are embedded in an agarose gel on a slide. Then there is the cell lysis. The DNA is liberated. The duplex DNA is unwounded. Then there is alkaline. You have to undergo alkaline electrophoresis, and the comets will be formed. How are these comets formed? There is a head and a tail portion. The head portion, okay, it represents the undamaged DNA, and the protruding tail portion represents the damaged DNA. So if the tail portion is large, it means that there is a very high DNA damage. So you can see this comet formation in your gel. That is when you run the alkaline electrophoresis. And the nature of the tail or the length of the tail will be directly correlated to the extent of the damage of the cell or the cell death. So cells embedded in agarose on a microscopic slide are lysed with detergent and high salt to form nucleoids. Electrophoresis at high pH results in structure resembling comets observed by fluorescence microscope. The intensity of the comet relative to the head reflects the number of DNA breaks. So this you can do, this kind of assay you can use for measuring any kind of genotoxic stress which a cell is facing. Another assay is there which is called tunnel assay. What we do here is the DNA fragmentation by fluorescing the free ends of the DNA, detects single and double strand breaks. Due to DNA fragmentation and strand breakage, the 3' hydroxyl termini becomes free. You then label it enzymatically with a modified nucleotide DUTP and deoxygenin, and then you add the anti-deoxygenin for signal detection. So TDT stands for terminal deoxynucleotidyl transferase, which is used to end label the 3' hydroxyl group exposed during cleavage process. But there are some limitations of this technique, that it is the sensitivity and the specificity. Lastly, we will spend maybe 2 to 3 minutes on what is meant by cell renewal. Renewal means the cell is renewed. The damaged cell tries to generate new cell, like whole body regeneration in Hydra. It can regenerate the foot and head after amputation. Organ-specific regeneration can be seen in the vertebrate liver. So, either to heal a wound or injury, or to replace the cells lost in maintaining tissue homeostasis. Stem cells can renew themselves. This is called self-renewal, maintaining the stem cell pool throughout the entire life. Self-renewal is similar to proliferation, as both depend on cell division. Embryonic stem cells, which are derived from the inner cell mass of the blastocyst, can generate any cell type in the body and hold an indefinite potential for self-renewal. And this ability is known as pluripotency, and such cells are known as pluripotent cells. The ability to differentiate into multiple distinct cell types is known as multipotency, and they are called the multipotent cells. So all these cells are capable of self-renewal. Now, how is this self-renewal regulated? It involves certain genes like OCT4 SOX2 nanog axis, then polycomb family genes, leukemia inhibitory factor, bone morphogenetic protein, so all of which suppress the expression of genes involved in differentiation. So all these genes, they are helping a particular cell for the self-renewal process. So to summarize, what we have learned today is, the first part basically we discussed about the cell death part. When we are talking of cell death, the apoptosis. Regarding the apoptosis, the significance, right? The difference between a dead normal, a necrotic cell, apoptotic cell. What are the extrinsic pathway of apoptosis, intrinsic pathway, coordinate these two. Discuss the series of events in an apoptotic cell. Okay, and the next part is that how will you identify an apoptotic cell? There are different assay, LDH assay, use fluorescent dye, comet assay, tunnel assay, and finally the self-renewal. How does a damaged cell, it renews, it leads to the formation of the new cell. So please go through this unit. I already said it's the unit number 16 in MSCZOO 001, Molecular Cell Biology. If you face any problem, write to us, feel free to ask questions, and we will be there to help you. Thank you all, and have a very, very happy learning.